In this video, we're going to talk a little bit about the Krebs cycle, a series of metabolic reactions which take place within the mitochondrial matrix. Here's our mitochondrial membrane. And they serve to process organic nutrients and provide for greater aerobic ATP production than might otherwise occur. And so if we go over here in the cytoplasm, we can see that we actually have our glycolysis, which is the processing of carbohydrates, right, from glucose to pyruvate, right, with a little bit of ATP production here. But our main aerobic output, at least, is these 2-pyruvate. Now, the 2-pyruvate can enter the mitochondrion and get converted rapidly into acetyl groups. Well, the pyruvate are 3-carbon molecules. We got 2 of those from our 6-carbon glucose molecule. Well, this pyruvate, the 3-carbon, got converted into the acetyl group, a 2-carbon, and so we gave off one of the carbons as carbon dioxide. And if you think about it, our nutrient molecules, all of our nutrient molecules, as they enter the Krebs cycle, amino acids, fats, or the carbon groups from glucose, the pyruvates, are comprised of carbon, hydrogen, and oxygen. And so our carbon and oxygen are being produced as waste products here and leaving, right? They're going to diffuse out of the cell and be exhaled, largely. Now, we could also have fat carbon or fat nutrients enter. They enter as acetyl groups. Amino acids can enter. They can actually enter as acetyl groups or at other places. We'll see that in some other figures. But the essential point is that we're adding carbon groups, many of them through acetyl groups, that enter the Krebs cycle, and they combine with oxaloacetate, a 4-carbon molecule, to produce, of course, a 6-carbon molecule, citrate. Now, the names here are not all that important to us. What we're trying to do is follow the carbons. And so we added a 2-carbon to a 4-carbon to produce a 6-carbon. And then our cycle begins. And along the way, we get these enzymatic conversions and the release of, as you see here, more of the carbon and oxygen, right? And if you think about it, here are our 2 carbons that came in with our acetyl group, and we get back to a 4-carbon group, and we can start the cycle again. The question becomes, where did the hydrogens go? And the hydrogens are really the end point, the end-all, be-all of this reaction, if you will, right? These are the metabolic gist. Right? And the hydrogen atoms end up attached to these 2 carriers, NAD, so in the form of NADH, actually more than one of them, and then as FAD, and there's another one over here, but also as FADH2, right? The carrier is FADH. And so we have hydrogen atoms, which is essentially an electron plus a proton being carried by these molecules. And they're carried out of the Krebs cycle from the nutrient molecules that entered, and they're being delivered to the electron transport chain. They're absolutely essential to the functioning of the electron transport chain, where most of our ATP is made aerobically, right? And so it's absolutely key that we deliver these hydrogen atoms to the electron transport chain to get all this extra ATP. Now you'll note there is one transition here within the Krebs cycle where the free energy drop is large enough that we can actually phosphorylate GDP to GTP, and that's the equivalent of ATP. So we actually do make a little bit of ATP directly, but again, it's a little bit like the ATP made in glycolysis, where it's not very much of the grand total if we can aerobically get the electron transport chain functioning and that maximum number of ATP from these nutrients.